The immune system's natural capacity to detect and destroy abnormal cells may prevent the development of many cancers. However, cancer cells are sometimes able to avoid detection and destruction by the immune system. Cancer cells may reduce the expression of tumor antigens on their surface, making it harder for the immune system to detect them. Express proteins on their surface that induce immune cell inactivation induce cells in the surrounding environment, microenvironment, to release substances that suppress immune responses and promote tumor cell proliferation and survival. In the past few years, the rapidly advancing field of cancer immunology has produced several new methods of treating cancer, called immunotherapies, that increase the strength of immune responses against tumors. Immunotherapies either stimulate the activities of specific components of the immune system or counteract signals produced by cancer cells that suppress immune responses. These advances in cancer immunotherapy are the result of long-term investments in basic research on the immune system, research that continues today. Additional research is currently underway to understand why immunotherapy is effective in some patients but not in others who have the same cancer. Expand the use of immunotherapy to more types of cancer. Increase the effectiveness of immunotherapy by combining it with other types of cancer treatment, such as targeted therapy, chemotherapy, and radiation therapy. One immunotherapy approach is to block the ability of certain proteins, called immune checkpoint proteins, to limit the strength and duration of immune responses. These proteins normally keep immune responses in check by preventing overly intense responses that might damage normal cells as well as abnormal cells. But researchers have learned that tumors can commandeer these proteins and use them to suppress immune responses. Blocking the activity of immune checkpoint proteins releases the brakes on the immune system, increasing its ability to destroy cancer cells. Several immune checkpoint inhibitors have been approved by the Food and Drug Administration, FDA. The first such drug to receive approval, ipilimumab, Yervoy registered, for the treatment of advanced melanoma, blocks the activity of a checkpoint protein known as CLA4, which is expressed on the surface of activated immune cells called cytotoxic T lymphocytes. CLA4 acts as a switch to inactivate these T cells, thereby reducing the strength of immune responses. Ipilimumab binds to CLA4 and prevents it from sending its inhibitory signal. Two other FDA-approved checkpoint inhibitors, nivolumab, Opdivo registered, and pembrolizumab, Keytruda registered, work in a similar way, but they target a different checkpoint protein on activated T cells known as PD-1. Nivolumab is approved to treat some patients with advanced melanoma or advanced lung cancer, and pembrolizumab is approved to treat some patients with advanced melanoma.